Good afternoon or good morning, depending on the time zone that you are joining us from. My name is Dr. Jenny Lawler. I'm Senior Research Director of the Water Centre at QEERI, and I'll be moderating this webinar today. So thank you very much, first of all, for joining. The title of our webinar today is the environmental sustainability of the agri-food sector. The agri-food sector is at a critical juncture, balancing the dual challenge of meeting the rising global food demand while urgently addressing environmental conservation issues. Experts from the Qatar Environment and Energy Research Institute, QEERI, and Tecnológico de Monterrey, Mexico, will collaborate to explore key topics today with a specific focus on understanding the intricate relationship between water, energy, and food. Our webinar provides an opportunity to engage in a crucial dialogue, sharing cutting-edge research and best practices. The overall goal is to pave the way for a more sustainable future in the agri-food sector, one that fosters the well-being of both humanity and the planet. So I'd like to introduce, first of all, the speakers that will be joining us today. Dr. Veronica Bermudez, Senior Research Director of the Energy Centre at Qatar Environment and Energy Research Institute, QEERI. Dr. Eduardo Sosa Hernandez, Research Professor at Tecnológico de Monterrey. Myself, I'll also be contributing a presentation. Also, Professor Roberto Parra Saldivar, Research Professor at Tecnológico de Monterrey. We also have joining us this afternoon or this morning, Dr. Kashif Rasool, Scientist at the Environment and Sustainability Centre at Qatar Environment and Energy Research Institute. We also welcome Dr. Elda M. Melchor Martinez, Research Professor at Tecnológico de Monterrey. And last but not least, Dr. Antonio Sanfilippo, Chief Scientist at the Energy Centre of Qatar Environment and Energy Research Institute. So without further ado, let me first do a few technical things. So first of all, we encourage you to ask questions. This is intended to be a dialogue, and we'll be very happy to answer any questions that may come from the audience. We encourage you to post the relevant questions in the Q&A tab on the lower side of the screen. So don't use the chat window if possible, use the Q&A tab, and then the questions will go directly to the panelists most efficiently. So without further ado, let me welcome our first speaker this evening, Dr. Veronica Bermudez, Senior Research Director of the Energy Centre at QEERI. And I will hand over to you, Dr. Veronica. Thank you very much.
Thank you very much, Jenny, for the very kind introduction, and welcome everybody to this webinar. So I'm going to share my screen without further delays and start. Oops. So here we are. So as Jenny told, I am Veronica Bermudez. I'm Senior Research Director of the Energy Centre here in QEERI. As you know, QEERI is a Qatar Environment and Energy Research Institute under Hamad bin Khalifa University, part of Qatar Foundation in Qatar. So I'm going to discuss a little bit about the latest trends, the challenges and opportunities that agrivoltaics brings in arid environment. And this work is being conducted together with my colleague, Dr. Juan López Garcia. So arid and semi-arid regions are span worldwide. When we think about arid and semi-arid regions, we usually think about deserts, but you would be surprised to know that dry lands are more than 40% of the land surface. The importance of these dry lands in food security is huge, because once again, we are not aware that 44% of cultivated areas are worldwide are in dry land. So this is putting a lot of pressure in water resources, and in particular with the ongoing climate change, it is putting a lot of pressure also in the specific specific challenges for the crops. Continuing with these challenges, so poverty, more than 1 billion people live in arid regions, and 100 countries are threatened by desertification. With the ongoing climate change, this is something that is going to be accelerated, and many regions in the world see this happening. As I was saying before, this is threatening water availability and also clean water access for more than 1 to 2 billion of people around the world. And considering that we use today the use of clean water is mainly devoted for irrigation purposes in the agricultural sector and also for landscaping, but this is another question. So land degradation. So due to all this desertification and all this ongoing climate change, 24% of the land worldwide is degrading. This is impacting 1.5 million. And why I'm saying that? It is because as I will show you later, agrivoltaic has an opportunity to help recover and recuperate land and to manage the degradation of land. And finally, another major concern, it is access to clean energy. From one side, it is related to the access to clean and safe energy that it is under the SDGs, Sustainable Development Goals of the United Nations. It is SDG 7, but also it is related to the fight and mitigation of against emissions of CO2 to fight against climate change. So there is a real need of going towards decentralized and more sustainable energy solutions. And also, as you may be aware, clean and safe energy is an enabler of other sustainable development goals. So, and to finalize, just as you have been listening to me, several words, climate change. So climate change is a question that it is not, we all know, agree now that it is not if happens or not, it's happening and it's getting worse. So this is just a global map of where are located mainly the arid and semi-arid regions and how they can expand worldwide. And we see that it is really well distributed around the world. So why I was telling all this to you? So because if we look into climate change and reduce agricultural, which reduces agricultural productivity from one side, and from other side required decarbonization of the energy sector and required land to install wind, to install solar. So solar is land consuming. From other side, we have the increasing in population, which from one side needs have an increase in food demand, and from the other side, increasing energy demand. At the same time, there is uncertainties in global trade. So this, from one side, has led to many countries to develop national plans for food security. In Qatar, we have these plans, but not only in Qatar, in many countries around the world. But from other side, we also see that there is a national interest worldwide to be self-sufficient in energy production. This is what we call energy security. So if we combine these two verticals, from one side, we need to have increased cropped areas. From other side, looking to solar, we need to have increased land areas, and we have an increase in land requirement for solar project. These both together bring us to agrivoltaics. So we are trying to solve different challenges with the same solution. So agrivoltaic concept, it is the double use of land for agricultural farming and photovoltaic generation in the same land area. So as I was telling you before, and you can imagine, due to all the challenges that I was enumerating, so agrivoltaic is been demonstrated and from a theoretical point of view and with several projects around the world that protects against high temperature and extreme weather condition. It's a protection for the crops, reduce the evaporation and increase the soil moisture, increase electricity production and efficiency. And one important thing it is that this electricity produced is close to the consumption point. So we are contributing to the safe and clean access to electricity in remote areas. From other side, contribute to increase the profitability and diversify the income of farmers, and has a number of benefits also in terms of water savement, improvement of the biodiversity, improvement of soil moisture. And this is from the crop point of view, but also from the photovoltaic and electricity production point of view, it improves the electricity production because it creates an ecosystem below the PV panels that allows to control temperature. And it helps to have a more sustainable way to provide food to in remote areas. From other sides, and you will see a little bit more of that, it has specific opportunities and specific benefits for semi-arid and arid regions, mainly related to the energy, food, and water synergy that is demonstrated for shade-tolerant crops, allows to have a longer harvesting season with higher product quality and water saving, and of course, look to improve working condition and is effective against desertification. So it brings a lot of elements that are very much related with sustainability and the mitigation of climate change. Of course, there are some challenges. So the challenging it is having agri-photovoltaic in desert environments. It is challenging. It is depending on the country. It needs to provide a huge number of electricity, depending on electricity consumption for the different countries to answer to the needs of energy security. It needs to work under extreme weather conditions and environmental conditions. So usually we see in arid and semi-arid regions, we see high temperatures with modules working at more than 30 Celsius. So this is with this we are losing efficiency, excessive radiation, high soiling. It means the accumulation of dust in the panels, high humidity, high UV. It needs to solve some limitation related to water scarcity for panel cleaning, so it is better to work with dry and so on. But there are solutions to manage this water. So some examples of how can be beneficial the use of agri-PV in dry areas. So we have seen in the literature, and we can see in the literature a number of agrivoltaic systems that show negative effect, that negatively affect crop growth. And in this case, this is due to shading. However, this negative effect can be reported to reverse, has been reported to reverse into facilitating effect on plant growth under drought conditions by reducing evapotranspiration. This is something that has been demonstrated for this tolerant shading crops. So in the figure here, we can see the effect of shading by photovoltaic panels on plant carbon acquisition and transpirational water loss in an agrivoltaic system. And plants shaded by PV will be less productive when sufficient water is available for growth in comparison to unshaded plants due to reduced photosynthetic activity. So this is this scenario A in the left-hand side of the slide. When water is scarce, plants will, however, profit from a reduced evaporative demand And in this case, it will contribute to reduce transpirational water loss underneath the panel. So this is scenario B. So in this case, yield will be more stable under shaded conditions, and in comparison to unshaded conditions. So we see that there is an advantage there. If we look into the figure in the right-hand side, so we can see here how irrigation, which is measured here in this figure as a soil moisture, is impacted by the agrivoltaic system when we compare it with traditional control installations on irrigation resources. So figure A and B in the right-hand side, as I was saying, show 30-minute average volumetric water content, which is related to soil moisture in the top 5 centimeters of the soil of the Agri-PV and control setting. C and D show differences between soil moisture in an agrivoltaic setting and in control plots, where positive values indicate additional moisture in the agrivoltaic setting. So for figures A and C, plot were watered every 2 days, and for figures B and D, they were watered every day. So you see that there is an improvement when we are working with agrivoltaic in dry areas. So I will skip that because I don't want to go much beyond the.
I'm going to give you your 1 minute warning, please, Dr. Veronica.
Yeah, exactly. So here just keep that the figures are related to the fact that agrivoltaic show a much better behavior in terms of crop for tolerant shade crops when it is related with dry regions than regions where we have available water. So, and it is seen either in the photovoltaic panels, but also in the cropping regions. So just to take away, as takeaways for you and just to finalize, so it is important, and agrivoltaic needs to be driven by farms needs, by cropping needs, and the first priority needs to be improve cropping and find the sweet hot spot where we have crops improvement and electricity production at the same, cannot be driven by the electricity production. So in this way, this is why I'm saying that the takeaway for us today, it is to look into agricultural aspects, light availability and light management, microclimate and plant, and more importantly, the most important thing is to select the most relevant and suitable crops. So there are a number of key performance indicators that are mentioned here, that if they are successfully achieved, we will achieve the right compromise and the right target of have high yield of crop production, while at the same time producing clean electricity for the farms and for local consumption. So thank you very much for your attention, and it was short, but I hope that you have enjoyed it.
Thank you very much, Dr. Veronica. We have a couple of questions from the audience, and we have a couple of minutes to go through them. So the first question that's come in from the attendees is there any particular type of soil that works best with agrivoltaics?
No, in terms of it is really crop dependent. So it is what works for your crop. What needs to be matched it is the crop, the environmental conditions, and the soil. What it is sure is that the soil will improve the biodiversity, and the soil moisture will improve with the use of agrivoltaic if we do not have, compared with non-agrivoltaic solutions.
Thank you so much, Dr. Veronica. A really interesting presentation. So we'll move on to our next speaker. Thank you so much. Our next speaker is Dr. Eduardo Sosa Hernandez. Good morning to you in Mexico, Dr. Eduardo. And as the first speaker from Mexico, I hope you can introduce the Tecnológico de Monterrey and tell us a little bit about the Tec de Monterrey before you start your talk. But we'll welcome you now to share your presentation and we'll proceed. Thank you so much.
I don't know if I am already sharing.
I think so. Yes, perfect.
Okay.
Thank you so much.
Thank you so much, Dr. Jenny, for inviting us in this webinar. Yes, of course, I will talk a little bit about Tecnológico de Monterrey and where we are working. We are part of this. Tecnológico de Monterrey is one of the largest universities, private universities in Mexico and Latin America, and is very focused on sharing the knowledge and also into creating the knowledge. And this is by the research and the process to generate new information. And as part of it, Tecnológico de Monterrey has launched 3 institutes dedicated only for research: the Institute of Future Education, the Institute of Obesity Research, and the third one, that is the one that we are participating, the 3 speakers from Mexico, is Institute of Advanced Materials for Sustainable Manufacturing. This institute focuses on the manufacturing process in Mexico because it accounts for nearly 20% of the gross national product. And as you know, we are a large area that also provides a lot of materials and products to the US, of course, the world. And with this in mind, we try to focus on material as part of everything that is being produced. And in particular, our unit within this institute is the bioproduction systems that leads Professor Roberto Parra. You will meet him later on. And our core is based on the biotechnology parts of the new materials, advanced materials. Without more introduction about Tecnológico de Monterrey, I will introduce the topic that I'm going to discuss with you today. And as part of this institute and Tecnológico de Monterrey, we have launched MARTEC. And in this talk, we want to talk about security for the food production chain through wastewater surveillance of pathogens, distribution, and monitoring. And MARTEC is a laboratory that focuses on water solutions. You can see our structure. Professor Roberto Parra is the founder of this laboratory, and co-founders are Professor Elda Melchor and myself. And we have the coordination of the laboratory by Doctors Alberto Aguayo and Orlando Rosa. And I will tell you a little bit about this laboratory history. We started monitoring emerging contaminants through a GCSO project, which where I met Jenny Lawler, Professor Jenny Lawler, through 2017 to 2019. And with this, we enter in this new area and work in different projects. And we established this laboratory in 2020, mainly due to the COVID pandemic, and we were focused on monitoring the SARS-CoV-2 virus. And of course, we designed this laboratory mainly based on the PCR detection methodologies. And we built our laboratory to detect, track, and quantify the outbreaks in Tecnológico de Monterrey mainly. And also from that we established some containment strategies and risk evaluations. And right now we are extending our horizon to new pathogens in the industry, in particular in the food industry. And here is the reason, no? There is a lot of biohazards in the food industry, and with this, it generates two main problems. One is focused on the population health, and you can see some figures from the World Health Organization. But one of the most shocking is that 33 million people die by this foodborne disease. That is mainly caused by pathogenic bacteria. Of course, there is other pathogens in the food chain, but in particular, this generates at least in the US 9.4 million deaths per year, and it's shocking. It's alarming because all this can be prevented and at least reduced in some manner. And the second part of the problem is the impact in the food industry. Of course, there is economic losses. In particular, direct losses arise to 20 billion US dollars. And of course, indirect losses derived by the new production of food, the containment strategies. Or sometimes you need to lose all the cattle, all the production, because part of it is contaminated, it has a disease, and it has implication in the food supply chain in different processes. Of course, the fattening process, there is working animals, and of course the productivity when you are producing new animals. Of course, there are pathogens that are mainly impacted on this, and of course there is a zoonotic risk to spread the animal disease to people. And of course, in this technology, we know that we can look at the whole supply chain as a different points to look for this type of pathogens, and we can identify where in this supply chain we can find the highest risk. Of course, the raw material, the production in the farms, the suppliers, the factories where you produce, and then there is the distribution, retail, and customers. Of course, not all these parts are able for us to detect, particularly for the customer. But previous to that, we have access to different points. In particular, in the production, we can see different spots where we can detect or take as a checking points for this analysis and monitoring. Another thing that we want to share with you is that as the World Health Organization, issues like the key foodborne disease hazards, of course, the hazards comes from the chemicals, but for the disease, for the pathogens, you can see bacteria, virus, and parasites. And of course, we desire to implement this PCR-based methodology to the whole process. And of course, with this, we focus on the pathogenics, mainly from the genetic material, but we can also detect the whole pathogen and the biomarkers by other techniques in analytical chemistry. In this particular talk, we focus on the genetic material, and we have the objective also to translate the gold standard from PCR to biosensors, because we need timely responses. We need more accurate, more online monitoring systems, but still we are on the way to produce this type of biosensors that are able to be applied in the industry. Right now, the MarTech workflow works like this. We identify different pathogens reported by literature or health institutions. And with this, we select in silico which part of the genetic material can be selected for its identification. Of course, we have the sequencing tools. If there is no report, we can also do some sequencing with some laboratories at Tec de Monterrey. And then with this identification of the highly conserved regions, we design some primers and probes for the detection of specific pathogens. Of course, we run in silico trials to make sure that we are working in the right direction. Of course, when we have a highly potential success rate in these probes, we synthesize or ask for them to be synthesized, and then we do all the through the tries in the laboratory with some samples, first model samples and then wastewater samples. And with this, we generate different tools to be applied in the food supply chain. Of course, right now we see three main parts where we can assess this type of diseases in the food supply chain, in the production, in the farms, in the slaughterhouses, in the process. There is a lot of water that we can, or points where we can monitor the pathogens, and of course in the food process industry when you have different post processing for the protein or for other parts of the animal. And of course, the idea is that we apply what we learned from the wastewater based surveillance for COVID, and we apply this now to the food supply chain. Right now, we have done some work already. We have these pathogens that are available for us to apply now to detect and to identify. Of course, there is influenza A H1N1 virus that affects mainly the swine livestock. Of course, in Mexico, there is a huge production of pigs. So this is important for us, and we know we cannot apply that in other parts, but we also have some pathogens for bovine or cattle livestock like Brucella abortus. We have also Listeria monocytogenes, Campylobacter jejuni, and cytotoxin E. coli. Of course, different pathogens affect different livestock, but at the end, there is also a possibility of zoonosis and all this transmission of these diseases to other animals. I know in Qatar you have some troubles in the food and farm industry by the rodents spread of diseases. It depends on the rodent and in the farm, but there is this problem. I know of it. And right now, all these pathogens have been, we have the probes and the primers, and we have identified at least, we have been able to identify up to 10 copies per liter as a limit of quantification. And well, I want to talk a little bit about another thing that we have been doing, this metagenomic analysis for the identification of interesting genes in the bacteria, in particular
Dr.
through
Duarte,
wastewater.
I'm just going to give you a 2-minute warning if that's okay. Thank you so much.
Thank you, Dr. And we do this metagenomic analysis to identify different bacteria that we are interested. In particular, we focus on antimicrobial resistant genes, and also in the ones that can produce biopolymers like PHAs. We have identified some of these strains in the wastewater, and we want to also take advantage of that. And as the last part, as as I was telling, we are trying to move forward to biosensors. And for that, we are right now developing a passive sample device that during the pandemic, we noticed that there is a challenge in the sampling, and there is a challenge also in the economics of the countries to apply this, mainly because of the logistics and the equipment that is required. So the first step is to develop a passive sample device, in this case an hydrogel, with the characteristics that allow us to trap the genetic material and then use it in the PCR. These are some of the preliminary results that we have. We have some formulations, and we have characterized the functional groups and also the soiling characteristics. We are finishing with the adsorption processes. And lastly, as some of the latest results, we are able to identify new pathogens, unidentified or unreported. In this case, we monitor the wastewater in Mexico for COVID, and we were doing application of clinical kits to identify in wastewater Omicron and Delta variants. And you can see the dynamics here from November to January. It changed a lot from Delta to Omicron, and also different mutations that are not reported in the literature. You see wild type that is reported, but then some mutations that are not reported declared as mutant in the RBT region of the gene that is for the spike protein. Thank you so much for your attention, and the content.
Thank you so much, Professor Eduardo. That's fascinating work, and the idea of this kind of biosurveillance in the water sector and its implications on the food sector is really novel. We have a couple of questions from the audience, please. So the first question that came in was, what's the kind of relationship between, and I'm just paraphrasing the question, what's the relationship between if you see evidence of pathogens in the water streams versus the likelihood of contaminated meat at the end of the slaughtering process and the processing and the packaging and everything like that? Is there any knowledge on the relationship or the likelihood?
Yeah, first of all, we need to establish a baseline. So we need to produce or analyze these slaughterhouse water streams in a periodic basis, so we can know what is the baseline. And from that point, when you can see a change in the pattern, particularly in a high or a spike, you can detect that this is an abnormality in the line, in the production line, and you have a potentially contaminated meat. Yeah.
Okay, and the second question that came in was, you had a list of different pathogens that you're detecting. Are they dangerous to have in foodstuffs?
Yes, well, some of them are dangerous, particularly the cyanotoxin E. coli is very dangerous for the human health. Other pathogens like the viruses, for instance, like influenza, you know, it has an impact on humans, but others are not really a hazard for us, but they represent a hazard for the production and for the yield that they can generate. And that's some of the differences that we need to take into account. Yeah.
Okay, that makes sense. Thank you. We did have some questions about your SARS-CoV-2, your COVID work, but I'm going to move on to the next talk because we can talk, you know, it's not really the topic of the webinar. So I will thank you very much, Professor Eduardo. Really fascinating. Really appreciate your talk. So the next talk on the agenda is actually mine. So I've already introduced myself. I'm Senior Research Director of the Water Centre here at QEERI, part of QEERI, part of Hamad bin Khalifa University. So I'm going to share my screen. Yeah, it was at the start. Okay, so topic that I'm going to talk about today represents a collaboration between QEERI and ExxonMobil Research Qatar. We are looking at the potential for non-conventional water resources. So as we know, and as we've heard earlier, the availability of fresh water in Qatar is very low. Our groundwater is significantly depleted, and the country's strategy is to move away from the use of groundwater for the agricultural sector. And for that, Qatar has looked very heavily at identification of other non-conventional water resources to serve the agricultural sector. So, for example, treated sewage effluent is now widely used in the irrigation of fodder for production of animal feed. So we've identified an alternative non-conventional water resource that, with the appropriate treatment, could potentially be used in the agri-food sector, particularly for fodder production. So what we've done is identified produced water from gas fields as a potential for treatment and reuse in this way. Oh, my slides are not moving. Okay, here we go. So the motivation for the research. So for those of you that aren't based in Qatar, I'll tell you a little bit about our situation here. So Qatar actually has 14% of the world gas reserves, and we're aware that Mexico is also an important gas and oil producing country. Produced water, if you speak to a lot of people in the oil and gas industry, they'll tell you that they're actually water producers with a side dish of oil and gas. The amount of water that's produced during extraction of fossil fuels is actually usually more than the amount of fossil fuel itself. So to give you an idea of the amount of water that's being produced, Qatar has a multitude of assets related to the oil and gas fields in the country, and each of those assets will have millions of meters cubed per year of water being produced along with the fossil fuel. And to give you an idea of the scale of that, our municipal water production, which in Qatar is virtually 100% from seawater desalination, on a yearly basis, that's at about 700 million meter cube per year. So if you look at the combination of water produced from all of the assets in Qatar, from the oil and gas assets, it's quite significant proportion of the water that is needed for the country. Qatar oil and gas fields are quite young, so the ratio of produced water to gas in Qatar's North Field, which is very young wells, is 1.2 to 1. So more water being produced, but in many parts of the world, and even in oil fields, it's even more. It can be as much as 8 barrels of water per barrel of oil or gas that's extracted from the ground. So really, really significant water stream with potential for treatment and reuse. So what's happening with this water typically at the moment, the Qatar situation is that from the gas fields, this produced water is injected into wells on land after some preliminary treatment to remove H2S. So the research objective for us is to look at this water and assess what we need to do to it to make it suitable for more beneficial reuse. So there are some parameters in the produced water that are quite challenging. So we needed to look at what different treatment methods could be used to make the water compliant with our standards for reuse. So as I mentioned, we have this important feasibility assessment. We're grateful for the participation of ExxonMobil Research Qatar for their collaboration in this project. Essentially what we look at is that potable water should not be used for non-potable uses. So water should be able to be made fit for purpose. So the quality levels should be made to fit the intended reuse. Whether it requires advanced treatment or not will depend on the final use case. So given that Qatar has a good history in terms of application of treated waters in the sector for landscaping and for irrigation of fodder, it has developed very good standards for the quality of the water that needs to be applied. So we looked at assessing the potential for this water to be treated to meet the standards. So some of the key things that need to be addressed, particularly for the Qatar situation, is the salinity. So the total dissolved solids that we see have to be below 2000 for irrigation, whereas for potable or drinking water quality, it needs to be down below 500. And just for context, the groundwater in Qatar is all salty. It's all what's termed brackish, so it's moderately salty. Typically anywhere from 8,000, 9,000 up to 15,000 parts per million or milligrams per litre salt, whereas the seawater around Qatar is over 40,000 milligrams per litre or parts per million. We also have contamination of this produced water with a variety of heavy metals and a variety of hydrocarbons, oils and greases, for example. So we needed to look at all of these elements as to its suitability for reuse. So when we assessed produced water from the Qatar gas fields, what we found was that the COD, the chemical oxygen demand, was challenging, out of scope, over 2,500 milligrams per litre. The total dissolved solids, which is a measure of the salinity of the water, was about 6,000 to 7,500 milligrams per litre, so over 3 times the allowed limit. Ammonia was quite high, and also hydrocarbons such as benzene, toluene, ethylene, xylene, and oil and grease were high. And it was also quite high in boron, various different heavy metals, and also monoethylene glycol, which is added to prevent freezing in the pipelines, particularly in the winter period. We also wanted to look, so you've seen in the previous 2 slides that there is a long list of compounds that you should assess to know whether the water quality is suitable or not. And as you can imagine, actually doing those assessments is quite challenging. It's quite tedious. It costs quite a lot of money to do that analytical work. So similar to our previous speaker, Eduardo, sometimes what you want to do is find a different way to analyze the water and make sure that it's suitable for use. So passive sampling is one way that you can reduce the burden, and another way that we've assessed in this project is to look at toxicity assessment to supplement full analytical testing. So we looked at our ultimate goal of testing: is this water toxic if it's applied to land? Is it going to be toxic to the environment, for example? And that can give us an indication if our treatment strategy is successful or not. So we implemented a hybrid treatment strategy, and this was informed by the learnings from Oman, where there is the largest constructed wetland for produced water treatment in the world. And this is operated by a company called Bauer, which we're fortunate enough to collaborate with, along with EMRQ. So they've designed for us a pilot scale constructed wetland system. It's small capacity, 24,000 litres per day in capacity. And a constructed wetland is a system that essentially has a lot of different processes going on to try and break down contaminants in the water. So it includes biological processes. So there will be active microbes and biofilms present within the constructed wetland material. We also have chemical processes, physical processes happening. We have oxygen transfer in the constructed wetland, which helps in terms of mineralization of contaminants. We also have evapotranspiration, where the water is evaporated from the wetland, and we have phytostabilization. So the plants are very important in terms of the operation of the wetland and removal of the contaminants. However, we could see from the work that had been done in Oman that the constructed wetland is not enough to meet all of the requirements. So we have looked at implementing a hybrid solution. We've tested a number of different technologies, including adsorption, coagulation, flocculation, and membranes. And we looked at the efficiency of pollutant removal from the produced water. So, as I said, the wetland treatment in Oman was not sufficient, particularly for TDS. It actually increased the salinity of the produced water as it went through the wetland. Also, boron, sodium, potassium, sulfate were challenging. The constructed wetland in Qatar will be installed actually next week. So these results are from Oman. So what we did was we got some samples of the water from Oman. We brought them here and we tested them. And we also took samples of the untreated produced water from Qatar. So these results are from Oman. So we took the raw produced water from Qatar, which similarly was quite contaminated. And we could see, first thing we tried was adsorption. So we have some novel adsorbents that were made in QEERI here, and we looked at could we remove some of these components. So we found that we were able to almost completely remove the BTEX compounds, the hydrocarbons. We had very good removal for some heavy metals such as aluminium and barium, but some of the other contaminants were quite challenging. So, and the TDS wasn't reduced at all. So we needed to explore some other options. We looked at coagulation flocculation with ferrochloride, and again we found that it worked well for some of the contaminants that were present, but not all. So in particular, again, the TDS was still high, it was still salty, we had ammonia issues, we had poor COD removal, and we still had chromium in the effluent. What worked very well was reverse osmosis membrane treatment. Which is really to be expected in terms of a physical removal of the various different contaminants. We did investigate nanofiltration as a potential option, and we didn't get terribly good results, particularly in terms of TDS removal was low, and for some of the other contaminants. But the use of reverse osmosis provided practically 100% removal of all the BTEX compounds and everything else that was of interest. So we were convinced that this would be a good option moving forward, and this was on the raw produced water. So what we wanted to do then was to utilize this as a hybrid nature-based, technology-based solution for treatment of the water. So we looked at taking the constructed wetland treated water from Oman and then testing that on the reverse osmosis. And what we found was that the permeate flux was much better when we used a brackish water reverse osmosis membrane. But very interestingly, what we found was that the constructed wetland pre-treatment meant that the flux or the productivity of clean water from the RO system was very, very much enhanced by about 35%. So I'm going to be my own timekeeper here. I'll just talk for a few seconds about the ecotoxicology tests that we employed. So this is to look at, is the water toxic without testing for every single compound, because there can be things that are toxic in different waters that we're not even testing for. You know, that's always something that can be a concern. So we looked at collaborators in Qatar University to set up part of this work. The animal model that was chosen for this work was zebrafish embryo. This is a very well established animal model that can investigate the effects of things that could potentially be toxic on human health. So we're grateful for the collaboration with the Biological Research Centre at Qatar University for these results. The zebrafish were the most sensitive to the untreated produced water. So this is evidence from the log COD value. So essentially at a very low concentration of produced water, the zebrafish embryos were killed very, very quickly. with the untreated produced water. So you can see that with the untreated water, the mortality in 96 hours for the zebrafish embryo at 120 hours post-fertilization, at an untreated water concentration of just 2%, the zebrafish couldn't survive. The copepods and the killifish, which are local organisms, they were much more resilient, but they were still quite susceptible to the toxic effects of the untreated produced water. However, when we looked at treating the produced water from the samples from Oman and also the samples from Qatar, we could see that with constructed wetland and membrane treatment and with the constructed wetland treatment on the water from Oman, that we were able to significantly improve the survival of the zebrafish embryos. So to summarize, the produced water, it is very challenging. There's a lot of contaminations over a wide range of concentrations. There is a lot of produced water potentially available that could be integrated into the water mix for the country. We tested a number of different treatments that could potentially provide good removal of the different contaminants, but the membrane treatment was the most efficient in terms of reducing the concentrations and particularly the salt that's present in the water. However, there is the potential to look at biosaline agriculture as a potential use for this water, having removed other contaminants with a combination treatment. The toxicity of the treated produced water was assessed using the zebrafish model, and it seemed to be quite successful in terms of removing the toxic effects. So the next step, we are, as I said, installing a 24 meters cubed a day constructed wetland in our partner site, along with the other scaling up of the other processes that we intend to try in the hybrid scale. And our next steps are looking at the techno-economic and feasibility analysis for the hybrid process. So I'm going to stop myself there because I can't see the questions unless I stop sharing the screen, and I will answer a couple of questions from the audience. Okay, so the first question from me. Does treating the water that comes out along with fossil fuel before injecting it back into wells consume a lot of energy? If yes, will it be a good option when compared to the energy needed for desalination? Okay, so treating the water that comes out, so the current treatment that is done typically in the oil and gas sector is just a pre-screening. So it's for essentially H2S removal and acidification. That doesn't take a huge amount of energy, but the injection itself does take some energy because it's deep injected underground. And the other thing is that there are concerns over the long-term stability of the well. So it's not just a case of the energy that's required for the process, it's also the potential long-term impact on the well health. So there could be knock-on impacts in 20, 30 years when we're still trying to valorize the fossil fuels. So this is one of the drivers for the research. Would it be a good option when compared to the energy needed for desalination? Yes, it is less energy intensive to use a hybrid process like this than seawater desalination. So the saltiness of this water is far lower. There's very little energy required for the constructed wetland. So the only main energy use there is some oxygenation of the wetland. And then obviously there's water pumping, but it should be far less energy intensive than seawater desalination. So it can be a good option to provide a source of water for the agricultural sector. So in the interest of time, there's another couple of questions in the chat box there, but hopefully we can come back to them during the discussion later on today. So I'm going to move on to our next speaker, which is Dr. Kashif Rasool from QEERI. So Dr. Kashif, if you'd like to turn on your camera and you'll be able to then share your presentation. Thanks, Dr. Kashif.
Everyone, good evening and good morning in other parts of the world. Let me open my presentation. Okay, can you see my P3 file?
We can see your screen, but your sound quality is a little bit muffled.
Okay, how is it now?
Yeah, please go ahead. Thank you very much, Dr. Kajan.
Okay, thank you, Dr. Jenny. I am Kashif Rasool, working as a scientist at Qatar Environment and Energy Research Institute at Hamad bin Khalifa University. So today I'm going to talk about a very important and interesting topic related to management and conversion of agro-industrial food waste into microbial, or also known as single-cell protein. So as we know, a diverse range of alternative protein sources that serve as a substitute for traditional proteins are being explored and produced, and these alternatives are usually developed because they are more environmentally friendly, they are more sustainable, and they can meet the growing demand for the proteins globally. And single-cell protein is among one of the alternative protein that can replace or complement the traditional animal feed. So this is the content of my today's talk. I will have a little bit time to introduce my project and how can be agro-industrial waste used to produce the single-cell protein, some of the results from our recent projects, and concluding remarks and acknowledgement. As we know, the world's population is steadily increasing, leading to higher demand for resources such as food, water, and energy. We know a large population requires more food production to meet nutritional needs, and meeting these demands lead to intensified agricultural practices. And this increased population and food production contribute to higher levels of the waste, including agriculture and food waste. According to a study, globally around 1.3 billion tons of the food is lost, which is equivalent to almost 30% of the total food produced. Only in the United States, approximately 700 million tons of the organic waste is generated annually, which translates loss of around 218 billion US dollars. So most of the waste, organic waste, which is produced, ends up in the landfills, which is ultimately converted to methane gas, which is a potent greenhouse gas and significantly contributes to the global warming and the climate change. So there is a need to embrace a circular economy approach where products and the materials are reused and recycled to reduce the waste while generating the value-added products. Agro-industrial waste refers to the byproducts generated from agricultural and industrial processes. These waste material can have various sources and may include residues from crop production. They can be a part of the processing material, like wastewater from different parts of the food processing industry. It can be a livestock waste, including the manure, and many post-harvesting waste produced from different agricultural activities. The valorization of agro-industrial waste involves converting these waste materials into valuable products or energy, which can minimize the environmental impact and promote sustainability. There are several technologies which are being employed for the valorization of these waste materials, including compost, anaerobic digestion, and several other biological processes which can produce the bioenergy, biogas, and biofuels like bioethanol and biodiesel. And also there are several microbial or biotechnological processes which are being utilized to produce different type of enzymes, pigments, alternative proteins such as single-cell proteins from these agro-industrial waste. So the topic that I am going to focus is about production of single-cell protein or microbial protein from agro-industrial waste. So as the world looks for more sustainable and alternative protein sources to meet the global or ever-increasing protein demand, single-cell protein represents a promising approach, I think, which is potential for efficient resource utilization and reduced environmental impact. Single cell protein, which is also known as microbial protein, is actually a derived cell of microorganisms, which can be used as a dietary protein supplement for animal and humans. There are different types of the microbes which are being applied for as a single cell proteins. These include algae and several photosynthetic bacteria, fungal and yeast, and several bacteria, including cumulithotrophs and methylotrophs. And all of these microbes can be cultivated using agro-industrial waste, depending on the type of the process that we are employing for this process. And the resulting protein-rich biomass can be used as a valuable source of protein. For example, here the table shows the nutrient composition of different types of the microbes, including fungi, algae, yeast, and the bacteria. So we can see here all of them are rich in protein. For example, bacteria has the highest protein composition from 50 to 65%, where all of them include a small amount of fat and some nucleic acids. So all of these composition of the nutrients from different type of microbes make these microorganisms a rich source of protein and other required nutrients for the growth of different type of livestock. A wide range of agro-industrial waste can be used for this purpose. For example, it can be a crop residue from post-harvesting. It can be a food processing waste from the industry. It can be also food processing waste or the dairy processing waste, or also manure or vegetable waste from different type of the fruits or the waste material. There are different type of fermentation processes which can be employed to produce single cell protein. For example, depending on the type of the microbes being used for this process, different fermentation techniques include use of submerged fermentation, which is a type of fermentation which is performed in liquid media, or it can be a semi-solid fermentation, itself a solid fermentation technique. So what is done actually in this process, the microbes are incubated with the type of different substrates, a carbon source and a reasonable nutrient source is provided for this one. And the microbes are cultivated with these different type of the substrates. The biomass which is produced is then post-fermentation process is involved in this one on the downstream, where it is separated from the other nutrient or the bioreactor sources. And then ultimately this replicated biomass is dried and used as a single cell protein, which is further formulated to be utilized as a feed source for different type of livestock, including agriculture, aquaculture, or poultry, or other hierarchy of the livestock. Single cell protein has already gained attention as an alternative protein source for animal feed, and so far there are several companies in the market which are actively involved in the production and commercialization of single cell proteins based products. Here I'm going to talk about several such products which are already commercialized in the market. For example, British Petroleum has pioneered the production of single cell protein, which is based on a yeast called Eurovia lepolitica, which shows an innovative approach to make single cell protein and use it as animal feed. There is another product available in the market known as Uniprotein, which is produced from a company called Unibio based in Denmark. Unibio is doing actually conversion of the natural gas methane to animal feed, and they are selling it as a brand name of Uniprotein. Similarly, on the same principle of converting natural gas into microbial protein, a company called Calista they are producing feed kind protein, which is also available in the market for the animal feed. The recent trend is using the waste materials, the materials which are causing environmental problem, and convert it to the animal feed. For example, a recent startup called No Nutrients based in the USA. They are trying to actually use carbon dioxide as a substrate for the microorganism, and they are producing single cell protein from CO2 and selling it in the market with the commercial name Nova Nutrients USA. So these are some of the microbial proteins or single cell protein which are already available in the market. However, current need or the current trend is to utilize
just a 1 minute warning, please, Dr. Kashif. Thank you so much.
So the current trend is to use the agro-industrial waste to or other waste material to convert these material to microbial protein. However, there are several issues related to use of these agro-industrial waste, including substrate variability, selection of suitable microbial strains, process optimization, and nutrient imbalance. What we are doing actually in our lab now to address all of these issues which are related to conversion of agro-industrial waste to protein is we are working on recently a project which is funded by QEERI and Ministry of Municipality. We are utilizing different waste streams selected from a local farm alternative. We are converting it to anaerobic digestion. By anaerobic digestion, we are converting it to the biogas, and the biogas generated from this process is being fermented by selective mechanocrops, and we are using it to produce a single cell protein, which is our objective is to convert it and use it to the animal feed and convert it back to the use to cradle approach and use it as animal feed. Another project that we are working on here is the conversion of agro-industrial waste to yeast biomass and use it as animal feed. In this project, what we are doing actually, we are optimizing the operational parameters. We are selecting highly rich protein producing yeast co-culture in our lab, and at the optimized condition, we are trying to produce single cell protein using semi-solid state fermentation process, and we are trying to produce a biomass which is enriched in protein. So far what we have achieved, we have achieved a co-culture of selected yeast strains, which has increased protein content in the dried biomass. And the biomass we have achieved is have a protein contents of almost 375% higher than that of the control. So this is the preliminary results we have achieved so far, and the objective is to scale up this process and use it as animal feed. In this process, we have our industry partner, Latin Agriculture Company, and we are future planning to install a pilot plant there to produce this single cell protein on the farm. So concluding remarks, we can have single cell protein as a high protein content with wide amino acid spectrum and can utilize it as a animal feed. This feed has an advantage that the growth of this feed is neither seasonal nor depends on the climate conditions, and it can be grown on the waste material from the agriculture and other type of organic waste. However, there are some challenges that we are currently facing in this one, which is substrate variability, presence of the pathogens and toxicants in the substrate material itself, and stabilization of the process. However, these challenges can be addressed with the careful substrate selection, substrate pretreatment, careful microbial selection, and bioprocess optimization. I would like to acknowledge our partner in this project. The funding receives QEERI and Ministry of Municipality in Qatar. Our industry partner in this one, a staffing company, and Ahmad University and Qatar University, who are collaborated in this project. Thank you very much, and I'm open for any questions.
Thank you very much, Dr. Kashif. It's an absolutely fascinating initiative. I'm just going to direct one question to you from the audience. They've asked, is it possible to use converted animal proteins, and are they approved as food for livestock in Qatar, for example, chicken feathers?
Yes, as I mentioned, there are several companies already who have commercialized microbial protein, and they are selling it worldwide. So definitely they are approved from their respective countries like FDA from USA and also from the agencies in the Europe. So they are available and can be used. In Qatar, we are trying to produce this microbial protein. We already have got US provisional patent on our this technology, and hopefully we will be able to commercialize it soon.
Thank you very much, Dr. Kashif. So I'm going to move directly on to our next speaker of the evening. And apologies, I did mix up the schedule slightly. So I'd like to welcome our next speaker, Professor Roberto Para-Saldivar from Tecnológico de Monterrey. So apologies for messing up the schedule, but welcome, Professor Roberto. And if you would like to share your screen, please do so.
Thank you very much, Professor. Yes, let me share the presentation. I don't know, let me put it in the show. Is it you can see my presentation?
It's perfect. Thank you so much. Gracias.
Thank you, Professor Lawler. First of all, thank you very much for this invitation. I'm very happy to be here. I feel that we seeing these amazing presentations, I feel like we have several things in common. First of all, you may not know, but Mexico is a semi-desert. Most of the land is semi-desert. 70% of the surface of Mexico is semi-desert. So we share similar risks and challenges. So the possibilities to collaborate with you are really amazing, and a lot of things. And from this, I think, again, from the Sustainable Development Goals, mainly the collaboration, I think, is what we can try to maximize in order to ensure the food security. So my talk is going to be related to the bioproduction systems for sustainable manufacturing. We're part, as Eduardo well mentioned, we are part of the new initiative from Tec de Monterrey, the Institute of Advanced Materials for Sustainable Manufacturing. And basically, we try to find the ways to ensure the sustainability of production, sustainability of large populations. So I think in that sense, we are very aligned, and I will start with this. We have this big problem. There's a lot of food losses and waste along the supply chain. There's a big problem. We see 60% of waste products, final products, are wasted because of some strategic key point was neglected. So we can try to implement this strategy in this work. We're going to present also some food waste management, as it was presented from Professor Khalif. He talked about that. No, waste is a very important source of new materials, and thinking about circular economy, it will be also a very important point to look at. And the group, what we are focused is on food waste management, edible coatings to increase the lifespan of fruits, single cell protein, also aquaponics. So we are going to talk about this in this presentation, give some conclusions, and of course questions and answers. This is what we see is in the base, in the production of food supply, production in total agricultural production, it has several key points that along the process are kind of eating the resources and basically are unable to recover in the way we are currently operating our system. So some of the waste is based on some seed that are not germinating, energy that is dissipated and lost in the environment, some use of feed to feed our sources. Some parts are not eatable, of course, and need to be recycled. Also, due to pests, this is the pest management, also another alternative to avoid the harvest loss that also observe. And this is before we go and harvest the products, and then after harvest, there are problems of the way we handle the fruits, for example, the quality that is required in the field. The way also post-harvest areas that had the handling and the losses, as I mentioned, in the transportation, temperature control, modify atmosphere, rooms to keep the ripening of the fruits suitable for the market. And of course, even though when the fruits are ready in the market, the processing and the retailer, fungi can spoil the products, and finally the real amount that is getting into consumer is just a little fraction of the whole resources. So thinking about the way we can reduce significantly this loss is taking account the white squares that are basically representing part of the material that is being disappearing or lost or wasted along the process. So, yeah, and in Tec de Monterrey, within the institute, of course, leading by Professor Arturo Molina, we face this inverted pyramid, where you show first we need to avoid the surplus of food in order to avoid production. That also has something to deal on the way we manufacture food, need to be just in time operation. Of course, it's very difficult as many parts are a big risk of losing or not getting enough food for society. But we need to find a way to make a suitable logistic for this. The reuse, in case it is reuse, because again, there is a big thing of food brands that they allow products visible in the market only for a very short period of time, and then even though products are in a good shape, they discard it and losing the opportunity to benefit somebody. So this is another opportunity. If we can redistribute use in a safe and of course following all the rules, it's fine. Some NGOs are working in this part, try to help people with disadvantage, economical disadvantage, to get cheap food. For example, probably some fruits that are not in the perfect shape, but still in a good way for human consumption. So this is another area, reuse. This is when the product is not suitable for human consumption, then a possibility to reuse and to transform into animal feed, as described in Hafiz's talk. Also, the recycle of materials, reuse, this is important, but this becoming, if we concentrate in the previous reuse, that will be the best. But if we are not able to reuse in some way for humans or for animal consumption, then the way we can recycle these products and have some added value onto these products. recycle in the case of we need to make a process with microbes in order to again recharge the material in order to have a benefit for society, recover of energy, and finally the least interesting area will be disposal. We can incinerate, we can get some energy, put it in a landfill and so on, but that will be the plan. This need to be reduced to the minimum and try to recover as much as we can at the top of the inverted pyramid, as the resources are more rich in that sense. Thermodynamically, you have a very high quality at the top, and the quality goes down as the time goes by and the process goes down in this pyramid. So prefer the top options. On that regards, our group is focused on food waste management, for example. We are working with some convenience stores, food retailers, in order to get these products and try to find opportunities, as I will describe. Other byproducts from agriculture, like the peel of oranges, can be used to produce polymers to enhance the lifespan of fruits. That is, well, I'm from Mexico, close actually from Michoacán. They produce a lot of avocado, and the avocado consumes a high amount of water. So if avocado get rotten, we are wasting water basically. So we try to find a shield that prevent fruits to rotten in the process. Also, we combine the aquaponics as another alternative, as it was presented by Dr. Veronica. The way of this agriculture related to electricity, energy, food production in a modular system should be implemented. So far, it's been neglected or not highly applied technology here. So it's a big opportunity as this aquaponic system I will describe here. Also, the production of single-cell protein. We're focusing these two technologies are based on pre-Hispanic technologies. For example, the milpa, the production of food and crops without soil. It was practiced since long ago in the Aztec culture, and the intake of microalgae as a source of protein also was a common practice. So a little bit thinking about how can we give the technology in order to enable this process to benefit a large number of people. So basically, this diagram describes the idea. Of course, we block the line, the arrow that goes directly to the landfill, and then turn it into something that can be a waste management plant to produce new food, and of course, extraction of molecules of high value. That's kind of an avenue, and also implement new source of technologies. Try to see what is happening in nature in order to speed up process like growing black soldier fly, fungi, or using microalgae as a source of proteins, lipids, and carbohydrates. Some vitamins also can be implemented, and of course, energy is one of the key products in this process in order to make it feasible for everybody.
Professor,
so basically, talking about mushrooms.
To interrupt you. I'm going to give you a 1 minute warning if that's okay, please.
Yeah. Well, I will go really fast. This is the work where we are doing with fungi as an alternative for protein. Also, black soldier flies for production of polyunsaturated fatty acid, omega-3, and using basically waste, and the extraction of polymers in a biorefinery concept in order to make covers, coatings for fruits to expand the life of shelf of these products. As an alternative, of course, represent a huge impact and an economical benefit for society, and the aquaponic system what integrates food and food actually, optimal use of water and so on. So this is what we are working. Of course, this is a diagram, but we are giving a lot of effort into the implementation of technology using artificial intelligence, of course, light and electrical panels in order to save energy. These need to be off the grid systems, so this is alternative. Some of the work that we carried out related to what we can do, produce fatty acids for human consumption using microalgae. Also the production of some proteins and that we can enable the production in large-scale bioreactors, and some coatings in order to, and some product that can be implemented along the process in our systems. So as a conclusion, we see when we share our ideas with all the other panelists, the use of agro-industrial waste generates an added value for products and that ensure the security of food, reduce gas emission, that is a problem, also enhance the lifespan of products, and of course implement circular economy to reduce. This is the team, and thank you very much. I don't know if I'm in time. Sorry if I extended a little bit more. Thank you.
No,
Thank you,
no,
Professor Jenny.
It's such an interesting program of work for the sustainability of the agricultural sector, and I can definitely see a lot of opportunities for collaboration there. Just in the interest of time, because we have two more speakers, I'm going to save the questions for now because they're saved actually, so I'll be able to come back to them to you. And we will move on to our next speaker of the evening, which is Dr. Elda Melchor Martinez. So Professor Elda, welcome. And if you would like to start sharing your slides, and you can turn on your camera and your microphone. So Professor Elda will be speaking to us about shrimp farming this evening, which is very much of interest to the food systems here in Qatar. Professor Elda.
Yeah, thank you, Professor Jenny Lower, for this introduction. I would like to start my presentation and acknowledge for this invitation. The topic that I will share with you is regarding on the valorization of the sargassum biomass. On this topic, we are working from the last 6 years ago, and we are exploring the potential of this natural resource in different applications. In this case, I will share the application of on the aquaculture sector, but we have some experience in the biorefinery of this natural resource, and we made some other applications on the bioplastics area and also for the bioremediation. Okay, to start with this talk, in the Mexican Caribbean coast, we are facing the massive influx of the sargassum. This brown macroalgae is distributed in several areas in the world, but in Mexico, we are facing the circulation of two species, the Sargassum fluitans and the Sargassum natans. Those are the species that we are facing as invasive or massive brown macroalgae in the Mexican Caribbean coast. And we know at the moment several environmental impacts that the biomass has for example, for the marine species or for the aquatic organisms, such as the corals, fish, and lobsters. But there are several reports also in which the decomposition of the sargassum biomass produce toxic gases and cause some affections on the respiratory system. And for Mexico, it's important to Mexico that the area in which we are facing this massive influx is a tourist area, and the people there are very affected with this issue, because some of the other activities or economical activities in which people are working on is fishing. And also the government and the hotel sector invest a lot of amount of money to clean the areas and keep the areas in more suitable property for the tourism activities. Okay, has a research unit in the Institute of Advanced Materials for Sustainable Manufacturing. I belong to the same group that Professor Parra and Eduardo are. And in this, with these opportunities to valorize the sargassum, we perform these biorefinery strategies in which we are exploring the potential to obtain biomaterials such as alginate, other applications. For example, for the agriculture sector, we are developing at the moment a biofertilizer. And in this talk, I want to share the results that we have in the sector of the aquaculture. With this natural resource, we start this investigation with a seasonal characterization. With this characterization, we consider 3 different factors: how the composition of the macroalgae can change if we collect in different seasons of the year, the place of collection, and the method of extraction. And we monitor the composition of the carbohydrates, proteins, and phenolic compounds, and other analysis and elemental analysis for monitor organic or inorganic compounds. And you can see here in the slide in the right side, you can see the 3 different locations that we use for the collection for this investigation. We saw that the season also changed the composition of the components, because there are some months on the year that are considered in the hottest season or in the wettest season. And in the next table, you can see our performance on obtaining carbohydrates, proteins, and the total phenolic compounds. And you can see here, we are comparing the my extractions and microwave extractions against the enzymatic extractions. And you can see 2 months, the May, that is considered the hottest month, and September, that we are facing here in Mexico, has a seasoning we are expecting more raining activities. Okay, and here you can see that, for example, if we compare in the microwave extractions, you can see that in the wettest month in September, we we were able to obtain more content of carbohydrate, proteins, and also we explore the potential to obtain these components from the fresh biomass or from the dry biomass. And we see that the fresh biomass is more suitable to obtain these components. And we see that the enzymatic extraction is not a convenient method to obtain these components, because in this enzymatic protocol we use some lacases obtaining from Pycnoporus sanguineus, and these lacases are not the most suitable option for this kind of biomass, because is the lignocellulosic content of this kind of biomass is lower compared with other kind of waste. Okay, and regarding the use of this biomass in the aquaculture sector, we published in this year this paper is regarding of the comparison of two biomass of sargassum of two different species that are circulating in two different areas in Mexico. We explore the potential of the sargassum collected in the Mexican Caribbean coast and other kind of species that are circulating in the Gulf of California. For this protocol, we have developed two new formulations, including this sargassum biomass, and we feed some shrimps in a control protocol or a biocide to reveal the potential of this biomass to use as a healthy ingredients in diets for swine farming. As you can see here in the left side, we use as a control a commercial formulation, and we develop two formulations, one with the sargassum from the Mexican Caribbean coast and the other one with the inclusion of fluor from the Gulf of California. In this protocol, we use two levels of inclusion, 2.5% and 5%. And we feed some juvenile shrimps. In this case, we use the Lithopenaos banamei. The protocol was performed during 35 days under controlled conditions, as you can see here. And before to start the protocol, we did the chemical characterization of the fluors. I want to mention that the inclusion of the biomass was after the pretreatment that was include dried conditions and after that grill the biomass. And you can see here we compare.
I just want to give you a 1-minute warning, please. Thank you so much.
Yeah, thank you. You can see here that we compare the chemical compositions of the two biomass of the two sargassum species, and you can see here that compared with the control, the sargassum, the new formulations based on sargassum has a very important results. We improved the growth of the animals, the shrimps, with this new formulation in these inclusions of level. It's reported in the literature that the lower inclusions is better for the growth rate, and because if we including higher, more than 10%, is not recommendable, because in this case we increase the viscosity of the intestinal contents, and is not suitable because change the digestibility of the new formulation. And you can see here other technical parameters. And also we analyze the digestibility with the monitoring of enzymes like amylase. And we see here that also the behavior of these enzymes. It was important to mention that we face we face that the enzymatic activity was higher than the control, and it is important to mention that it's related to better absorption of the new formulations. Okay, I want to finish this talk saying that we are collaborating with other universities, with the University of Ghana and University of Southampton and University of York, to develop educational material to let all the people know about the situation with the sargassum and how we can valorize through biotechnological tools. Okay, conclusions, and I want to take to all my team and collaborators, and I will be very happy to know about your questions.
Thank you so much, Professor Elda. That's absolutely fascinating and really interesting to see that you're already collaborating in the MENA region. Just because we're short on time, I'm not going to go to questions, but we can have follow-up. We will take the questions from the audience, so we'll be able to direct them to the panelists and get back to the questions afterwards. So apologies for that, but thank you very much. So I'll move directly to our next speaker, Dr. Antonio Sanfilippo from QEERI. Thank you so much, Dr. Antonio. And Dr. Antonio will be telling us about optimizing crop yields. So if you'd like to share.
Jenny, thank you. Let me share my slide. Let's go to the beginning. So the topic of our discussion here today is sustainability in indoor farming, and particularly with reference to energy in a hot desert climate. So few introductory remarks about the issue that we're dealing here. It's basically scarcity of fresh water and arable land, make food production very challenging in hot desert climate, which include, by the way, Mexico too, as you can see from the map there. And Qatar is a very good example. As you can see from the pictures, the center and the left of the slide, very little groundwater suitable for food irrigation, and extremely little land for agriculture. But only 1.8% of the country is arable. Indoor farming obviously provides a natural solution to these challenges, and particularly with hydroponics, you avoid the issue of no soil needed, and also, because a controlled environment approach to agriculture, you can use a lot less water and fertilizers, and aquaponics obviously offer additional benefits, as has been remarked by previous speakers. Obviously, there are challenges here, a higher operational complexity, and also higher energy requirement for temperature control. To address these problems, we started a project about a year and a half ago, funded by the Ministry of Municipality and Qatar National Research Fund, and includes University of Malaysia, Pahang, Agri-Food, which is the major food producer here in Qatar, and a research station of the ministry. And the goal here is to use Internet of Things technology and AI, artificial intelligence methods to simplify operational complexity, optimize the use of resources, and also perform economic analysis to understand, where to go from the analysis of optimization of resources. Here is a little example of what we do in terms of installing, engaging Internet of Things technology. So basically installing all bunch of sensors, soil sensors, environment sensors, cameras in the greenhouse. These sensors are hooked up to microcontrollers, which then communicate via radio frequency to a gate where the information that is sent to the cloud or where it's essentially analyzed. So it's a way of doing this sort of efficiently in terms of cost, because obviously you need to put several layers of sensors in the greenhouse. Maybe depending on the size of the greenhouse, you need 3, 4, 5, 6 lines of these sensors, and obviously you don't want to use internet to communicate inside the greenhouse. Okay, so this is the system that we are developing, and it is really about dynamic predictive assessment of crop productivity and the ensuing sort of economic advantages. So we start in the greenhouse, collecting sensors, as was shown before, and also outside. And then what we do, we have a greenhouse digital twin. And what the digital twin does is essentially reproduces the working of the greenhouse. It works as, like a virtual greenhouse. So we do is like, for example, every week, every month, you instantiate the model with whatever the data you have from the greenhouse, and then you use those data to do the simulation to get the crop output at the end of the cycle, whatever it is, 90 days, 60 days, 9 months, depending on the assumption that you make. And then you essentially generate a variety of crop scenarios using little variations of a given selected resource interval. So, for example, if we're looking at temperature, the optimal temperature for tomato might be 22 centigrade Celsius, and you generate all a variety of scenarios up to 35, which is the essentially the top level temperature that tomatoes can endure. And so you end up with all these kind of scenarios, and that's how you do optimization. And then you apply economic analysis on the results of the optimization. I just want to give you a better feeling how this works out. This is like a view of the greenhouse digital twin. You make a number of assumptions in fashioning this digital twin, essentially to reproduce the same working of the greenhouse. And the digital twin essentially works through a number of equations that model photosynthesis, heat, ventilation, and how these impact plant growth. We also have, for the purpose of energy, we also have a component which estimates energy consumption. And you can see here, in terms of energy use, what would be, where the energy consumption would come from: cooling, air circulation, and so on and so forth. Okay, optimization, as I was saying, what we're doing in optimization is essentially creating all these different scenarios. And then in this particular case, we're using a multi-objective genetic algorithm to select out of the thousands of scenarios which are the best one, which offer the best trade-off between energy use and yield in terms of crop productivity. Obviously, we're doing here this for energy, but you can do it for any other resource like water, nutrients, and more interestingly, you can do it for all of these resources put together. So, you know, you end up with a very complex multidimensional graph, so to speak, where you have different optimization choice of all these components, of all these resources. Finally, you know, you do your optimization. Very interesting here to see that, you know, out of the many thousands of scenarios, these 5 ones are the ones that offer the best trade-off, the so-called Pareto front. What is very interesting is that, you know, you with a little bit more of energy, you know, maybe 2,000 kilowatt hours, you get to a good, you know, over 24 kilos per square meter. But then, you know, you can add a little more. You can add 10,000 kilowatt hours, and you only have a very little improvement, maybe, you know, 100, 200 grams per square meter. So you might want to say, well, you know, it might be more useful to produce a little less crop, but to use a lot less energy. And ultimately, that is what the techno-economic analysis will decide, and that's what we're doing here. You taking into account all the operational and capital costs and the revenues that might come from the sale of tomatoes, with different quantities and at different prices. And then you can see what essentially the baseline is in terms of breaking even and making a profit. You know, obviously, this is different with different scenarios, optimized scenarios you may use. So again, you know, here is the summary of the workflow as we saw it. You start with information from the greenhouse and the environmental side. You do your modeling and simulation to kick off optimization, and finally, techno-economic analysis. So in conclusion, food production in hot desert climate is challenging, no doubt, but these challenges can be addressed in terms of introducing indoor farming. But indoor farming comes with its own challenges. And the work that we're doing shows that Internet of Things technology and artificial intelligence automation can actually address and hopefully solve these challenges. And with this, I'd like to finish off and thank you all. And you know, obviously, if you have any questions, I'll be glad to answer them.
Thank you so much, Dr. Antonio. Really, really interesting, and I can immediately see some lovely potential to extend the study towards the added value of the waste products from the production process that can be incorporated into the techno-economical modeling. You know, there's a really, really nice opportunity there to put some of the different projects together in that way. So I'd like to, Dr. Antonio, if you'd like to stop sharing your screen, and we'll just go to wrap up the webinar because we're slightly over time, and I'm conscious that we're live streaming to the JOA Expo 2023. So I hope that the audience, both online and at the expo, have enjoyed the evening. It's been a fantastic whistle-stop summary of some of the amazing research that's happening both in Mexico and in Qatar. You know, really ways that research and development can contribute towards the sustainability of the agri-food sector in the desert, semi-desert, arid environment where we have a significant lack of water and challenges for food sustainability in the two countries. So I'd like very much to thank all the speakers this evening. It's been an absolute pleasure to speak to you. and I'm looking forward to follow-up meetings with you to discuss further our areas where we could potentially collaborate and further share the knowledge between our two countries, and a true example of globalization. So thank you very much to the team, both from QEERI, HBKU, and from the Tec de Monterrey, Mexico, and live streaming at the Doha Expo 2023. Delighted to have you. Thank you very much, and we will bring the webinar to a close. Thank you so much.